Reading forests, Yunnan. Identifying an evergreen oak. Oaks form interbreeding hybrid swarms, and yet distinctions are somehow maintained. Names only open the mystery. 17. Flying spores. All of this is, of course, speculation. My college is Shungping Shu discussing Matsutake evolution. Landscapes and landscape knowledge develop in patches. Matsutake Shiro, mycelial mats, model the process. Patches spread, mutate, merge, reject each other, and die back. The hard work and the creative, productive play of science as well as emerging ecologies happens in patches. But one might also wonder what moves beyond them. For Matsutake, there are also flying spores. Both in forests and in science, spores open up our imaginations to another cosmopolitan topology. Spores take off toward unknown destinations, made across types, and, at least occasionally, give rise to new organisms, a beginning for new kinds. Spores are hard to pin down. That is their grace. In thinking about landscapes, spores guide us to, in population, heterogeneity. In thinking about science, spores model open-ended communication and excess, the pleasure of speculation. Why spores? Koji Iwase first started me thinking about spores. We were having lunch in Kyoto with Shiho Satsuka and Michael Hathaway. The tape recorder wasn't on. I was curious about why Matsutake is so cosmopolitan. How did it spread among the northern hemisphere? Dr. Iwase is generous with foreigners and willing to guide them, so he mentioned that the stratosphere is full of fungal spores. At those high altitudes, they blow around the earth. It's not clear, he continued, how many of these spores survive to germinate in distant places. Ultraviolet radiation kills, and most spores are viable only for a short time, perhaps a few weeks. He didn't know if a Matsutake spore could survive to germinate on another continent. Even if it did, he explained, it would have to find another germinating spore. Without fusing, it would die in a few days. Still, over the course of millions of years, one might imagine that spores could spread the species. 1. Interview 2005. There is something about the stratosphere that inspires airy dreams. Imagine spores circling the globe. My thoughts took off with drifting spores, chasing their protagonists across eons, across continents. I took my questions to mycologists here and there around the world, chasing their thoughts too through the stratosphere. I found a cosmopolitan science of speculation about origins and the makings of kinds across space and time. Unlike the discontinuous patches of applied forestry, the science of Matsutake spatiation is not patch-like. There are strong winds of international consensus about methods. The materials, mushroom samples and DNA sequences, circulate across borders. Individuals and sometimes lab develop stories, bits of expertise, and even biases. But there are no schools, no patches. All this work is off the clock. No one gives out grants to study the eon-crossing travels of a mushroom. Scientists turn to those questions out of love, and because the methods and materials are there. Perhaps one day the combined results and speculations will lead us, like spores, to something new. They reason. For now it is just the pleasure of thinking, the spore-filled, airy stratosphere of the mind. What are these materials and methods that circulate? Henning Knudsen showed me the University of Copenhagen Botanical Gardens fungal collection, of which he is a curator, too. Interview 2008. Type specimens are stored here, drawers and drawers of folded envelopes, each guarding a dried fungus. When a new species is named, the neighbor sends a sample to the herbarium, and those species become the type for that species. And those specimens become the type for that species. Researchers from around the world can ask to see the type. The herbarium sends the original material. The herbarium system emerged with a Northern European passion for identifying plants, which also resulted in Latin binomial names. It was a feature of European conquest. It also created the basis for transnational communications through the circulation of specimens. Researchers around the world know species through type specimens collected in herbarium. Dr. Knudsen does not think Matsutake spread through spores in the stratosphere. It's just too improbable that they would find mates. Instead, their distribution followed the forests. They spread together with the trees. This took a long time, but across the northern half of the earth, many species spread, ever so slowly, together. Some, such as Boetius edulis, may have spread across the top, from Alaska to Siberia. But the homogeneity of northern species is overstated. Many species that used to be seen as uniformly found across the global north turn out to be different species, he said. 3. See Henning Newton's and Jan Vesterholt's taxonomy. Fungi Nordica, Copenhagen, Nordsvamp, 2012. The rejection of uniform cosmopolitan species draws not from the circulation of herbarium samples, but from a revolutionary new technology, DNA sequencing, which offers a new way to define species. Mycologists examine particular DNA sequences, example the internal transcript spacers, ITS region, that tend to be conserved within species, but show variations across them. Jean-Marc Monclavo, Dr. Knudsen's counterpart at the Royal Ontario Museum in Toronto, explained that more than a 5% divergence in the ITS sequence indicates a new species. 4. Interview 2009. DNA sequencing does not reject the materials and methods of herbaria. Most comparisons across species use herbarian samples, but there is a new material here in circulation, the DNA sequences themselves. Databases have made it possible for scientists around the world to consult DNA sequenced by others. The simple precision of DNA sequencing has taken the scientific world by storm. There is no alternatives. It seems so powerful that scientists keep making up questions based on the availability of this answer. Of course, there are still pockets of difference. Dr. Moncalvo explained that as recently as the 1980s, Chinese mycologists had trouble communicating freely with Europeans and North Americans. One Chinese mycologist sent him samples of fungi hidden between the pages of reprints. As a result of isolation, he said, Chinese taxonomies are strange. Internationally, there are no rules for naming a genus. The first name is a Latin binomial, so Chinese taxonomers have added China to genus names, assembling Cynoboletus instead of Boletus, and confusing foreign counterparts. Furthermore, they recognize species indiscriminately. They claim to have 21 species of oyster mushrooms in Yunnan, but there are only 14 species recognized in the world. Tiny morphological differences are given too much attention, but this is changing now, he said, as young scientists with international training take over. What do these materials and methods tell us about kinds? Species has always been a slippery concept, and DNA sequencing, despite its precision, has not made it easier to handle. Classically, species boundaries are defined by the inability of individuals on each side to mate and produce fertile offspring. That's easy enough to figure for horses and donkeys. They mate but do not produce fertile offspring. What about fungi? Dr. Moncalvo walks me through what it would take to find out 2 different fungal strains or species according to this definition. You would need to germinate 1 single spore of each culture, get those spores to mate, somehow force them to produce a mushroom, then get its spores to mate and produce mushrooms. For a fungus such as Matsutake, for which no one has succeeded in producing a single mushroom culture, and whose spores don't even germinate if alone, such experiments are hardly worth conceiving. Besides, Dr. Moncalvo added, imagine the hapless graduate students who devoted a dissertation to finding a species boundary of even the easiest to handle mushroom. Where would he or she get a job? Where would he or she get a job? All this matters in getting to know Matsutake across its diasporic locations. 20 years ago, there were many, many species of Matsutake scattered around the northern hemisphere, with more emerging constantly as scientists found them. Now there are just a few, and growing fewer. This is not because of extinction. DNA sequencing in the ITS region has allowed scientists to argue that most of these kinds of Matsutake are really just one kind, Trichoма matsutake. T. matsutake now appears to spread across most of the northern hemisphere, not just across Eurasia, but into North and Central America. Only Trichoma magnivelare, the matsutake of North America and Pacific Northwest, is continuing to stand clearly as a separate species, and even it is very close in its DNA signature to T. matsutake. 5. The name Trichoma caligatum, also T. caligata, is used for several quite different fungi, some counted as matsutake. See prologue, number 11. The precision of DNA sequencing, which allows Such determination also undermines confidence in the species as basic category for understanding kinds. I first met Kazuo Suzuki, now president of Japan's Forestry and Forest Products Research Institute, when new results were coming in about the identity of China's oak-loving matsutake, at that time called Trichoma zangdi 6. Interview 2005. In Japan, matsutake are associated with pines. Only false matsutake are found with broadleaves. The association between matsutake and conifers seem part of its species definition. DNA studies showing the close relation between China's oak-loving matsutake and Japan's exclusively pine-loving ones caught researchers by surprise. Dr. Suzuki brought his younger colleague from Tokyo University, Dr. Matsushita, to our meeting to tell me the news himself. His examination of the ITS sequence had shown no species difference between oak and pine lovers. 7. See also Norihisa Matsushita, Kensuke Kikuchi, Yasumasa Sasaki, Alexis Guerin-Leggett, Frederick Lapeyrie, Lumin Vairio, Marcello Intini, and Kazuo Suzuki. Genetic relationship of Trichoma matsutake and T. nauseatum from the northern hemisphere based on analysis of ribosomal DNA spacer regions. Michael Science, 46, 2005, 1996. But Dr. Suzuki, who had worked with matsutake for many years, did not accept this finding as the whole story. It depends on what question you ask, he explained. He told me about Armillaria root rot, a complex of species in which clear species boundaries may not be relevant. Armillaria root rot spreads across whole forests, stimulating boasts of the largest organism in the world. Differentiating individuals becomes difficult, as these individuals contain many genetic signatures, helping the fungus adapt to new environmental situations. 8. Peabody et al. Haploid vegetative mycelia, cited in Tracking Interlude number 21. Species are open-ended. When even individuals are so molten, so long-lived, and so unwilling to draw lines of reproductive isolation. Armillaria root rot is 50 species in one species, he said. It depends on what you are dividing species for. I remember the discussion vividly. I was at the edge of my seat. Dr. Suzuki was treating species in the way cultural anthropologists treat their units, as frames that must be continually questioned to retain their use. The kinds we know, he implied, develop at that fragile junction between knowledge-making and the world. Kinds are always in process because we study them in new ways. This makes them no less real, even as they seem more fluid and beckoning of questions. Ignacio Chapala, a forest pathologist at the University of California, Berkeley, was even more adamant that the idea of species limits the stories we can tell about kinds. This binomial system of naming things is kind of quaint, but it is a complete artifact, he told me. You define things with 2 words and they become an archetypal species. In fungi, we have no idea what a species is. No idea. A species is a group of organisms that potentially can exchange genetic material, have sex. That applies to organisms that reproduce sexually. So already in plants, where out of a clone you can have change as time goes by. You have problems with species. You move out of vertebrates to the cnidarians, corals, and worms. In the exchange of DNA, and the way groups are made, are very different from us. You go to fungi or bacteria, and the systems are completely different. Completely crazy by our standards. A long-lived clone can all of a sudden go sexual. You can have hybridization in which whole big chunks of chromosomes are brought in. You can have polyploidization or duplication of chromosomes, where a completely new thing comes out. You have symbiotization, the capture of, say, a bacterium that allows you to either use the whole bacterium as part of yourself or use parts of the bacterium's DNA for your own genome. You become something entirely different. Where do you break down the species? 9. Interview 2009. To compare different kinds of matsutake, Dr. Chapala used herbarium specimens as well as fresh samples and sequence ITS region DNA, but he refuses to imagine his results as fixed species. You start getting these groupings that you can only name relative to each other. You can't call them a species. In the old taxonomic approach, you say, This is my ideal. It's completely Platonic, and everything is going to compare as a missed approximation to that ideal. Nobody will be the same at this, but you compare and see how close they are to this ideal. If it becomes too different, by whatever measure, and the measures are completely arbitrary, you say, oh, this must be a different species. To avoid a false scientific cover, he speaks of Matsutakis as all the varied kinds that enter the Japanese trade. His study did, however, find distinct genetic groupings by region. This means, he said, that genetic materials are not freely exchanged across those regions. If you see good patterning, if you see good separation, that tells you that there is not much exchange between these groups. These data show that cross-regional exchange of spores is unlikely on a regular basis. One down for the long-distance travel of spores, but other possibilities have just become more thrilling. How then do kinds travel? Dr. Chapello, working with his colleague Dr. Garbellotto, has a story to tell about Matsutaki's travel. 10. Ignacio Chapello and Matteo Garbellotto. Phylogeography and Evolution, Matsutaki and close allies as inferred by analysis of ITS sequences and AFLPs. Ecologia, 96, number 4, 2004, 730, 741. The Eocene ancestral population, he argues, developed in North America's Pacific Northwest, where T. magnivillare continues to associate with both broadleaves and conifers, in resonance with that broadleaf-loving ancestor. The rest of the Matsutaki group jumped to conifers and has followed conifer forest ever after the northern hemisphere. When conifers retreated into refugia, Matsutaki followed, especially with pine. Wherever the pine forest went, Matsutaki went too. Migrating across the Bering Straits, Matsutaki colonized Asia and then Europe. The Mediterranean Sea blocked gene exchange between southern Europe and North Africa. Populations on each side are independent extensions of the vast Eurasian trek. Meanwhile, Chapella and Garbellotto imagine that southeastern North America is colonized by Matsutaki from the rich pine oak refugia in Mexico. Their story was shocking, in part because at the time they published, most people thought of Matsutaki as an Asian species complex. After all, only Japanese and Korean loved Matsutaki and thought of it as their own. How could it be a North American mushroom that came late to Asia, even if millions of years ago? Chapella and Garbellotto date the separation of T. magnivillare, another Matsutaki, as having occurred 28 million years ago, with the rise of the Rocky Mountains. Indeed, not everyone argues with the story they tell. This is an open-ended field. Dr. Yamanaka of the Kyoto Ecological Institute argues for a Himalayan origin for Matsutaki. 11. Interview 2006. Katsuji Yamanaka, The Origin and Speciation of the Matsutaki Complex in Japanese with English Summary. Newsletters of the Japanese Mycology Association, Western Japan, Branch 14, 2005. Many new species came into being with the rise of the Himalayas, which forcefully threw old kinds into new environments, stimulating difference. At the time of the Chapella and Garbellotto research, the evidence of host differentiation among Matsutaki in southwest China was not readily available, at least in California. It turns out the Chinese Matsutaki associate not just with conifers, but with Quercus as well as Castanopsis and Lethocarpus, which find their center of species diversity in the Himalayas. Dr. Yamanaka reminds me that major broadleaf host of North America's T. magnivillare is ten oak, and the only non-Asian Lethocarpus, 12. Manos et al., worried about how an American Lethocarpus might exist, have moved tin oak to a new genus, Lithocarpus. Paul S. Manos, Charles H. King, and Sung Hong O. Phylogenetic relations and taxonomic status of the paleoendemic Fabaceae of Western North America. Recognition of a new genus, Lithocarpus. Madrono, 55, number 3, 2008, 180. Might this be a clue? Dr. Yamanaka found Matsutaki Shira in China, associating with both conifers and broadleaf hosts. He argues for Himalayan origins, based in part on the sheer variety of mycorrhizal arrangements in the area. Diversity is often a sign of time and place. Yet, even newer research has shown that southwest China's Matsutaki are not particularly genetically diverse, at least in the ITS region, most commonly sequenced by researchers. They are a whole lot less diverse than Japanese Matsutaki, which everyone argues to be latecomers on the evolutionary scene. But this does not mean they are a newer population. Zhangping Xu of Canada's McMaster University suggests that Chinese Matsutaki just fill up more of the available space than in Japan. 13. Interview 2009. This saturation, he points out, can lead to longer living clones with less genetic competition. The stress of industrial pollution might also lead to genetic competition in Japan. Southwest China is far less industrialized. Diversity is not just about time and place. Dr. Xu brings back the question of spores. Many mushroom species are widespread. They are opportunistic. Whenever there is food, they can survive. Dispersal is not such a significant barrier for most of them. He brings up the panspermia hypothesis, which posits that spores are everywhere, traveling even in outer space. For most microbial species, you can find them everywhere. Dispersal is not the barrier, it's whether they are able to survive in these environments. He jokes, it's like Chinese now, they are everywhere. If there are business opportunities, you're probably going to find Chinese. If there's a small town, you'll probably find a Chinese restaurant. We laugh together. He talks of how well spores are dispersed. For many species, there are limited genetic differences among populations from very different geographic areas. One example is the bacteria in our mouths. He says that bacteria in the mouths of middle-class urban Chinese are vastly different from those of their peasant neighbors, but just the same as the bacteria of North Americans on a similar diet. It is the environment, not the location, that matters. For many fungi, too, he confirms, dispersal is not the problem, especially since humans emerged. There's a new thought. Humans? Dr. Xu is not the only one who thinks that human trade and travel have dispersed fungal spores. Dr. Moncalvo finds that very significant, although he disagrees with the idea that spore clouds are everywhere. Mushroom populations are restricted and well defined. The same morphology on two different continents is usually separated by genetic distance. There is exchange through spores, he argues, but it is occasionally not constant. But exchange may be much more common now because there is more trade and more travel. For example, Amanita muscaria was transferred to New Zealand in the 1950s and is now spreading. It is not even out of the question that matsutake spread across the Atlantic with human contact. There are a lot of Scots pines here. Scots pine is a major North Eurasian matsutake host, but it is not native to the New World. Canadians, they still have the queen on the coin, right? So they think the pine seedling that are coming from Her Majesty's garden must be better quality than native pine. He shakes his head in mock horror, but it is a serious point. Perhaps matsutake traveled to eastern Canada on the route of pine seedlings. Dr. Moncalvo does not dismiss the possibility of spread without humans, but he does think the spread must be recent, because eastern North American matsutake are so very similar to Eurasia ones. And he adds, shockingly, who knows which way the spread went, especially if we find the two species, Western Americans T. magnivelare, cosmopolitan T. matsutake, coexisting in the Central America and possibly in the southern Appalachians. That might be the origin. One, T. magnivelare, has been stuck on the west coast. The other, T. matsutake, has moved. That is something a phylogenetic study should be able to tell. Phylogenetic. How might both species have come to Mexico, I ask? I was a southern refugee during the glaciation, he explains. It's a well-known phenomenon. The southern limits of oaks and pines are the mountains in Central America. You can't find them in South America, and you find them with altitude. When it gets cold, everything moves south. When it gets warm again, they move up to a higher altitude. 3,000 meters in Mexico is like sea level here. This can also explain some shuffling. Populations will grow back from local refugee, but they are not salmon, swimming back to the stream in which they were born. There is no reason that one gets one way or the other way. It's the ecosystem that moves. It's not the fungus that moves. It's the ecosystem that moves. No wonder humans move so many species without meaning to. We create new ecosystems all the time, and it's not just humans that change things. I rather think it can sometimes be events. Dr. Moncalvo explains to my repeated questions about how kinds spread. There's something that many people cannot grasp. That time frame is huge. The tectonic separation between the southern and the northern hemisphere is 100 million years. So we find different species in the southern hemisphere and the northern hemisphere. Australia is a great example. So people say, oh, they separated 100 million years ago. But it's not true. Now that we have molecular data, we see it's incorrect in most cases. They are isolated, but there is sometimes transfer. But the transfer is not all the time. So we don't have something homogeneous. That could be one transfer per million years. Or per 10 million years. That transfer could be anything. It could be a tsunami wave starting from the Philippines and crossing the equator. They don't usually cross the equator, but in 100 million years and carried on the top of the wave, some soil and some wood with some animals hanging. It could also be wind. It could be anything. Once mycologists thought that southern and northern hemisphere mushrooms had been isolated for 100 million years, but DNA sequences now show that this could not possibly be true. For Amanita, for example, there are many groups with north-south ties, rather than just a single hemispheric dichotomy. Assumptions about slow and constant mutations in place are being displaced by attention to unusual events, indeterminate encounters. How do kinds emerge, then, in local populations? Dr. Xu explains it. Scale matters. One cannot use the same tools to study cross-continental and local diversity. The ITS region of fungal DNA is fine for studying big blocks of regional difference, but it is useless to study local populations. There are a completely different clump of DNA is needed to judge the variations that separate one group from another. Dr. Xu found that single nucleotide and polymorphisms, SNP, are good for population differentiations. 16. Anthony, 14. Zhangping Xu, Honggu, and Xu Liangyang. Single nucleotide polymorphisms in the ectomycorrhizal mushrooms. Tricholoma matsutake. Microbiology 153, 2007, 2002, 2012. With this tool, he studied matsutake populations in China, finding little genetic difference between oak and pine-loving matsutake, but a significant geographic separation across sampled regions. Most important, perhaps, this separation added evidence that sexual reproduction is important in matsutake populations. Spores rise again. In the world of fungi, this is not at all self-evident. Fungi propagate through many mechanisms, and sexual reproduction through the making of germinated spores is just one. A good deal of fungal propagation is clonal. Some clones, including those of the famous Armillaria root rot, are large and very, very old. Fungi also propagate through asexual spores, which are produced in times of stress. With their thick walls, they withstand hard times to germinate when better conditions return. For some species, sexual reproduction is absent or rare. For matsutake, however, the evidence suggests that sexual spores are important. This is investigated by examining the genetic composition of clonal patches. Are they mutating independently or exchanging genetic materials? For example, do you find more genetic diversity in older forests rather than in younger ones? Where would you expect a founder effect rather than free spore dispersal? For matsutake, the answer to this last question is yes. Spores appear to be exchanged among patches of mycelial growth. 15. Anthony Amon, Sterling Keeley, and Matteo Garbalotto. Forest stage correlates with fine scale spatial structure of matsutake mycorrhizas. Mycological Research 113, 2009, 541 to 552. This seems, however, landscapes features can block the exchange of spores. Researchers found that ridges, for example, block genetic exchange among matsutake populations. Anthony Amon, Matteo Garbalotto, Zheng Shenghong Feng, and Sterling Keeley. Isolation by landscapes and population of the prized edible mushroom Trigolama matsutake. Conservation Genetics 2, 2010, 795 to 802. This seems familiar enough, but don't relax. Matsutake does something strange and wonderful that can turn your idea of sexual reproduction upside down. It was another meal, tea this time, in Tsudabaka City with Hitoshi Murata of the Forestry and Forest Products Research Institute and Matsutake World's team member Liba Fayer. 17. Interview 2006. I was so excited when I understood that I spilled tea all over my tray. Dr. Murata had been studying the genetics of matsutake populations. It was a painstaking process, since matsutake is not an easy research subject. Figuring out how to get spores to germinate was itself a problem. They germinated, he found, in the presence of other matsutake parts, for example, mushroom gills. This suggested that spores might germinate best on living shears, that is, mycelial mats, including that of the parental body that gave rise to the mushroom. 18. According to Dr. Murata, matsutake does not have a somatic incompatibility system to restrict matings. See Murata et al. Genetic Mosaics, cited in chapter 16, number 9. And what happened then, when they germinated? This is where his research reveals something wondrous. Matsutake spores are haploid, that is, bearing only one series of chromosomes, rather than paired sets. We might expect them to mate with other haploid spores, thus making full pairs. They do. Human eggs and sperms join that way, but matsutake spores are capable of something else. They can join with body cells that already have chromosomal pairs. This is called daimon, mating from the prefixes for 2, the number of chromosomes copies in fungal cells, and 1, the number in the germinating spore. 19. Haploid nuclei in fungal body cells may not combine until production of fruiting bodies. Meanwhile, producing cells with 2 or more nuclei, each with 1 copy of the chromosomes, the dye refers to fungal body cells with just 2 haploid nuclei. It's as if I decided to mate with, not clone, my own arm. How queer. The spore brings new genetic material into the shiro, even if it is shiro's offspring, because the shiro itself is a mosaic, a combination of multiple genomes. Even emerging from the same shiro, different mushrooms might have different genomes. Even emerging on the same mushroom, different spores might have different genomes. The genetic apparatus of the fungus is open-ended, able to add new material. This adds to its ability to adapt to environmental shifts and to mend internal damage. Evolution in one body, the fungus can discard less competitive genomes to pick up others. Diversity emerges right there inside the patch. 20. For an opposing view, Si Chunlong Lian, Maki Narimatsu, Kazuhide Nara, and Tazuo Hogetsu, Tricoloma matsutake and natural Pinus densiflora forest. Correspondence between above and below ground genets, associations with multiple host trees, and alterations of existing ectomycorrhizal communities. New Phytologist, 171, number 4, 2006, 825 to 836. Dr. Murata explains that he was able to ask these questions because of his unusual background for a mycologist. He was originally trained in bacteriology. Most mycologists come from botany, where they see one organism at a time, or ecology, where they see interactions across organisms. But bacteria are too small to care about one at a time. We know them in patterns and masses. As a bacteriologist, he knew about quorum sensing, the ability of each bacterium to chemically sense the presence of others and to behave differently en masse. From his very first studies of fungi, he found quorum sensing there. In fungal mosaics, each cell line can sense the other, forming mushrooms in unison. By examining fungi differently, a new object came into sight, the genetically diverse fungal body, the mosaic. Mushrooms come with genetically diverse spores, mosaic bodies, chemicals sensing that creates communal effects. How strange and wonderful the world. I struggle. Isn't it time to come back to patches and compatible scales and the importance of history? Shouldn't I return to multiple rhythms, the tempos through which patches emerge in both landscape and science? But how happy it feels to fly with spores and to experience cosmopolitan excess. For the moment, the reader must make do with hasty conclusions. Spores vitalize matsutake populations through adding new genetic materials. Mushrooms produce many, many spores, and only a few of them germinate and mate. But it is enough to keep populations cosmopolitan and diverse. Some of that diversity is within the parental bodies that produce the spores. No one fungal body lives self-contained, removed from indeterminate encounters. The fungal body emerges in historical mergings with trees, with other living and non-living things, and with itself in other forms. Scientists speculate about open-ended questions, including the evolution of spread and matsutake. In a spore-like way, most of those thoughts never make a difference, but the few that do can revitalize the field. Cosmopolitan knowledge develops out of historical mergings, with research subjects living and non-living, and with itself in other forms. Patches are productive, but there are also spores.